Welcome to the poster presentation entitled Environmental Conditions Shape the Nature of a Minimal Bacterial Genome. My name is Magdalena Antczak, and even though I will be presenting this poster today, this research has been done with the collaboration with my supervisors, Martin and Mark, from the University of Kent in the United Kingdom. In 2016, scientists from Craig Venter Institute designed and synthesized first minimal bacterial genome. This genome had 473 genes and 530,000 base pairs. And these were only the genes essential for survival and for growth. And this minimal bacterial cell grows in a nutrient-rich dish. The question that was immediately asked was, what do these genes do? What are the functions essential for life? The group used three methods to find the functions of those genes, and they were unable to find confident functions for 149 of them. In the process of annotating the genes, they split them into 5 functional classes according to confidence and specificity. And the 2 classes with the least confident and specific functions are considered to be the proteins with unknown functions. They also assigned functional categories to those genes, which represent biological processes they are involved in. Our objective was using a combination of results from an extended scope of 22 different computational methods, compare the functional groups between each other and see if there are any differences between the proteins with known and unknown functions, and finally predict the functions of the proteins in the unknown group to increase the understanding of the minimal genome. We benchmarked our method by applying it to the proteins with known functions and comparing the results with the initial annotations. We found that our predictions for 92% of the proteins, our predictions agreed with the initial annotations. However, our approach has increased the confidence of these functions with an average of 13 methods supporting them. When we compared the functional groups between each other, we found that the proteins of unknown function are depleted in orthologs and known protein domains. However, they are also enriched for disorder and transmembrane domains. And when we looked at what part of the protein we could model confidently, we found that for only 9 proteins from the unknown group, it was possible to find a confident structural model. We assigned the functions to 133 of the 139 proteins with unknown functions, and for 66 of them we were able to find more informative functions. The predicted functions cover multiple areas such as protein synthesis, cell division, and transport. In fact, we predicted 24 new transporters. When we look at more general functional categories, we see that 50% of the genome is responsible for preserving and expressing genomic information. And I would like to show you one example. Here's a protein that was initially annotated as a putative membrane protein with generic confidence. We did in fact predicted 13 transmembrane helices. However, we also found that fire to matched the structural model to 26 templates with transport functions. And also we predicted many gene ontology terms related to transmembrane transport. And finally, Interpro revealed that this protein belongs to a major facilitator superfamily. So we ended up with a function transmembrane protein, likely a cation transporter from major facilitator superfamily. As a conclusion, we would like to postulate that each minimal genome depends on the environment it grows in. So each of them has two sets of genes, one essential prerequisite for all bacteria, and another one, an additional set of genes that enables life in a particular environment. So with the increase of nutrients in the environment that the minimal cell grows in, we observe decreasing number of genes responsible for metabolism and biosynthesis, but the increasing number of genes responsible for transport. And in the end, I would like to thank everyone for listening, and ISCB for inviting me to talk today, and everyone in my lab involved in this project.